All right, hello everyone. My name is Sean Elliott. I am a professor in the Department of Chemistry here. I have various other affiliations. You can find me on Twitter for as long as that exists until it goes up in a flaming heap. You can check out our website here if you are so interested. We are thinking about the molecular world today for the next 4 minutes at least, from a bioinorganic chemistry perspective. From the standpoint of bioinorganic chemistry, there's a significant intersection between the energetic sciences, thinking about energetic processes in the world around us, and global geochemistry. So with respect to that, our group has two major research efforts that focus in this sort of intersection space of bioorganic chemistry, enzymology, and sustainability sciences. First of them involving the fixing of carbon, and the second involving the breaking of molecular nitrogen. So with respect to fixing carbon, the microbial world has solutions to our problems. They're very good from the standpoint of taking CO2 and transforming it into useful things. The chemists that we take our inspiration from are microbial chemists. So various kinds of microorganisms, methanogens, magnetotactic bacteria, acetogens, all have the capacity for transforming carbon dioxide into useful molecules. Some of those useful molecules are arranged in a reverse version of our own TCA cycle, something that for those of you that shudder inside, if I mentioned the Krebs cycle, that is the same idea, except going in the reverse. Instead of producing CO2 and reducing equivalents that you then use for making ATP. See, I know I can see the people inside sweating if I make you try to commit your memory to the Krebs cycle. In this case, we're doing the opposite. We're using reducing equivalents that are coming from a redox active carrier protein called ferredoxin, and we're capturing carbon dioxide in two steps, one of them involving an enzyme called P4, and one of them involving an enzyme called OGOR. And so our group is interested, again, at this molecular level of trying to understand how these catalysts operate, how they might be tuned by nature to preferentially engage in CO2 reduction instead of the opposite oxidizing direction. So what does that mean? That means that sometimes we're interested at the level of enzymes in developing structural views of how these enzymes operate, how they engage in their electrochemical behavior, how they move electrons into and out of their active sites. We measure redox potentials. We do a lot of electrochemistry in my research group, trying to understand the nature of these thermodynamic couples and how that translates into the ability of the enzymes to operate as a robust catalyst. In a more applied direction, we work in collaboration with Gordana Dukovic's group at the University of Colorado at Boulder, generating these as light activatable O4 enzymes, taking these enzymes, absorbing them onto light harvesting nanocrystals, and turn them into light inducible reagents. And so being able to transform with light and carbon dioxide, carbon into useful materials. With respect to 1 minute left, fantastic. In respect to the opposite problem of thinking about taking something instead of capturing it, breaking it apart, we have to have a picture of a cow to think about the nitrogen cycle. Nitrogen is the world around us. We want to be able to make nitrogen where we want to make nitrogen available in the form of ammonia where we need it to be. And once again, there are microbial processes at work into converting the various forms of elemental nitrogen, sometimes that are problematic, sometimes that are very useful. In this context, my group is mainly focused in a process that allows us to acquire nitrogen in the form of ammonia that would be bioavailable from nitrite. And so this again involves a different class of enzymes, so-called nitrite reductases, that can interconvert to nitrite into ammonia, a 6-electron redox reaction using heme cofactors. And so we are interested in these pathways of electron transfer. And again, reading this out in the form of electrochemical analysis, or in this case, in the cyclical tammetry experiment, we're sweeping the potential, measuring the flow of current as current washes through the system, and we interconvert nitrite into ammonia, which you could think of as a sensor, or you could think of as an element of a fuel cell, or as a means of generating ammonia in situ if needed. And with that, I'll hand things off to the next speaker.